All right, I'm greeting you all, my friends. My name is Alex Surnov, and this is my first attempt to give a public talk online in English. Previously, I have been giving my talks in Russian, and for a long time I refused switching the language because I'm sure there are a lot of much better narrators than me who are native speakers in English and talk approximately about the same subjects, the same topics, immunology, cell biology, and give basic introductions to public audience about these questions. And I didn't want to compete with them. However, recently I introduced another presentation that I made on coronavirus infection that is currently spreading across the world. The infection that is called SARS-CoV-2. And several of my friends asked me to translate the same talk from Russian to English and present it for a broader audience. I'm personally not very sure that so many people will watch it, and probably the negative impact that this presentation will make will not get so vastly seeded and scattered. So I decided to accept this challenge and present my materials for English speakers as well. I hope it will be interesting. However, before starting talking about the subject itself, I would like to give you a disclaimer. I'm not a physician. I did receive a PhD title in immunology, but by education and by current occupation, I am a biologist, and I have no right whatsoever to work with patients and to give formal medical advices. So everything that I say is just a private opinion of an independent individual who has access to publicly available literature sources and who has some experience in searching for appropriate literature. Basically, this is what my talk is gonna be consisting of. I will give you a short review of relatively conservative and not that rapidly changing information that can be found in literature on coronavirus SARS-CoV-2 and the disease that it causes, namely COVID-19. So I guess after this introductory words we can proceed to the main subject. And first of all, let's take a look at the viruses themselves, and I will refresh your understanding of what do we mean when talking about viruses. So in contrast with bacteria, helminths, other complexly organized parasites, viruses cannot be really treated as purely live objects because they do not exhibit signs of life when exposed outside of the host body. Outside the host body, a virus particle is not a living cell whatsoever. It's rather just a mixed crystal of proteins, maybe lipids, and some nucleic acids on which the genetic information of this virus is recorded. However, and by the way, it's incapable of replicating itself when it stays alone. In order to start its life cycle and to precede the life cycle, it has to intrude into a host cell, and then everything it does, it just translates the information recorded on its nucleic acid, whatever it is. It can be either RNA or DNA, and translates this information in such a way that it takes under control the internal machinery for protein making and for nucleic acid making that is pre-existing in the cell, and redirects this machinery for creating more and more virus particles instead of for performing the intrinsic needs of the cell. And then the cell, as I said, produces a lot of viruses, a lot of virus particles, gets exhausted, and probably just dies of exhaustion, or bursts and spreads out the newly born particles, or it just keeps surviving in this infected mode until it gets killed by the immune system of the body. So what else do we have to know about viruses? Again, in contrast with bacteria, which are relatively complex cells, for viruses, usually there is no known specific therapy. By this I mean that large numbers of bacterial species can be successfully destroyed, successfully cured by the same antibiotic. But the instances when the same antiviral drug works against multiple different viruses are not that common. Usually a drug is designed for a certain virus and is ineffective for most of other viruses. Nonetheless, instead, for many viruses and virus-caused diseases, vaccines are effective, or better saying, effective vaccines have been developed. And the examples are rabies, smallpox, poliomyelitis. So vaccines, generally, every vaccine works as a boost for immune system. The immune system of the host body gets treated with attenuated or inactivated causative agents of the disease, and later the immune memory cells are formed, which take over and start a massive response once the body is exposed to the same pathogen again in the future. And this new response that is mediated by the memory cells is so effective that the symptoms do not have time to occur in this case. Okay, so here are a little bit of introductory information about viruses, and let's take a look at which viruses have been known there in recent 2 decades, let's say. Recent media stars in the virus world. So in roughly 2003, this respiratory syndrome, acute respiratory syndrome created or induced by coronavirus appeared in Southeast Asia, and it got its name SARS, and we all know about this. There was an outbreak of so-called atypical pneumonia, SARS-CoV. Later, a few years ago in 2012 or whatever, A similar virus occurred in Middle East and it was called MERS-CoV. They both belong to the family of coronaviruses, the same family that gave us the current virus, the virus SARS-CoV-2 that is currently spreading across the world. And also there were two massive outbreaks of, or worldwide outbreaks of flu. Flu viruses belong to another family, not to coronaviruses. There was bird flu H5N1 and swine flu H1N1. And some more local but also dangerous flashes of infection were associated with the viruses Zika and Ebola, and you all know about them for sure. However, these guys were rather local. These were successfully treated, and SARS appears to be, unfortunately, worldwide infection. It looks like the presence of the of my face image here takes so much of memory and capacities of the computer that it's better to start talking only with the presentation. I hope you can forgive me. I hope it doesn't really matter whether my face is visible or not, because who am I to look at me all the time? So let's transit it to slideshow mode and keep recording. Yeah, does it? It's recording. Good. By the way, while I was thinking about these technical issues, I also came up with one more disclaimer that I have to pronounce. My speech right now does not represent a formal opinion of the institute where I work. I also even will not mention in which place I work. I have to repeat that I act here only as a private individual and share only my personal opinion. All right, let's go. Let's continue. The viruses, the current virus that causes this COVID-19 disease is very closely related, very similar to SARS-CoV from 2003. And here there is a scheme of the viral particle from this paper in Nature in 2005, and this scheme depicts the viral particle of SARS-CoV number 1, the one that appeared in 2003 and spread out in 2004 or whatever. But the current one looks pretty similar. It also has a lipid, meaning fat, membrane, or not membrane, a layer, better say a layer outside of it, where a few different proteins marked by different shapes here are anchored, and some of these proteins form such a crown-like structure for which the virus got its name, coronavirus. Inside the particle there is single-stranded RNA on which the genetic information of this virus is recorded, unlike us, who keep our genetic information on DNA. This virus prefers RNA for this purpose, and the RNA molecule of the virus is embedded in the nucleocapsid protein. Once the virus gets in the lungs, this outer domains of the proteins on its surface attach to certain proteins on the lung cells, and then the virus intrudes into this lung cells. So herein, when I show references like this, it means that the full bibliography and the full description of those papers and materials will be present in the end, and I will give you time to go through it. Even although I am not a physician, I'm not a doctor, I still think that these literature sources can be helpful to physicians, or at least to physician students who may listen to me right now, if they are not too bored. So what we can extract from these papers about the current coronavirus? Well, first of all, it seems to be stable outside the host body for around 3 days. And also, it's pretty contagious. It's really effective in airborne transmission. It gets spread out with cough, sneezing, just with any kind of contact like this. And please, by the way, don't hesitate to go to WHO, World Health Organization webpage, see the full PDF report about coronavirus. It's pretty interesting. It's downloadable and it's available for everyone who is curious. And the same I also can say about CDC, Center for Disease Control in America. And the corresponding reference will be given later as well. So what's next? So what does happen when this virus gets into the cell? Well, there is a paper published in the Lancet, a very prominent medical journal. I'm deeply apologizing for this. The Russian title has, by whatever reason, not been converted. Essentially, it says the progress of coronavirus infection, COVID-19. That's literally what it says, and I apologize that I didn't translate it previously. So here is the paper in Lancet, and the good about this paper is that it has a long table with all medical parameters from the patients in China who have been detected with coronavirus infection recently. By recently, I mean in this year and this season, the coronavirus 2019 and 2020. For physicians who listen to me can be particularly curious about this if they need to compare their patients, for example, to what has been published previously. So, as I said, the infection occurs in lungs. Not occurs, but the virus gets seeded in the lungs and starts the infection. And the inflammation that follows the infection onset is easily detected by X-ray scan and all other kind of tests for it. The symptoms can be pretty delayed, can be undetected for as long as 24 days. However, sometimes they start instantaneously, and the average duration of asymptomatic periods of COVID-19 infection is 3 days. So it's between 0 and 24 days, but at average 3. And the hallmark of this infection, the business card of this virus, what is the most prevalent in most of the infected individuals, is cough and fever. And those two symptoms outnumber, for example, such things as runny nose, sneezing, vomiting, diarrhea, and something like this. Those symptoms also can happen, but are much rarer. So, all right, here I did translate the name. Another paper which summarizes known facts for infectivity, or for the potential of this virus to infect other individuals, and for its longevity is pretty interesting. And from this paper we can get that during his or her disease course, an infected person at average, I repeat, infects 2 to 3 more other healthy people. This is the pace with which the disease spreads right now, and the authors emphasize that even the person who is infected but is still asymptomatic, who does not cough and does not notice that something is bad with himself, is already contagious and already can spread the disease throughout the population. This virus, despite it is pretty rapidly spreading, fortunately is not that likely, I should say, to lead to death, although I don't want to underestimate its danger. Here I need to emphasize that by any means this virus cannot be treated as not dangerous, cannot be ignored, cannot be taken unseriously, and so on. No, it still can harm, and it still creates a lot of unwanted loads on economies on different countries. So about its potential to kill people, what we can say, well, if we just simply divide the number of dead people, the number of dead people to the total number of ill people, this ratio should be flipped. It's the number of dead divided by the number of ill, but doesn't matter. So we all understand what happens. If we calculate the percent of deaths among the ill people, in case of coronavirus 2019, it's going to be just 2.2%. In case of the previous SARS from 2003, it was 9.6%, and in case of the virus MERS, the virus from Middle East, it was the whole 34%. However, those guys were reported to be not as contagious as the current one. The same paper the same paper that I previously referred to, the paper from Lancet, identified some risk factors that are associated with poorer forecast about this virus infection. That's unfair. I again didn't translate the title, but again, it's the risk factors. You know what it is. The risk factors that these authors listed in their big master table are hypertension, diabetes, coronary heart disease, and these are the 3 main ones. The hardest progression of this infection, the hardest outcome is detected in people of older age or people with some chronic diseases that require permanent maintenance, like God forbid oncological diseases, people with suppressed immune system or whatever. Well, all these, the full list of these risk factors you can find in the corresponding publication, but 3 main ones are hypertension, diabetes, and coronary heart disease. All right, now I hope you, my friends, did not get scared. Let's take a look at which kind of drugs are available for treating this condition. And the drugs are, the list of drugs is taken from this paper here. Here the drug name is listed vertically, and this is the official name of the substance. And then as of beginning of March 2020, the status of different drugs is also different. Some of them are just proposed. They are potential drugs. They haven't been they surely have not been approved by controlling governmental organs, and also probably they did not undergo all spectrum of necessary tests to be counted as safe and effective. One substance here has, as the authors write, improved the condition of one patient. Okay, let's watch what this drug does next. I believe the researchers are testing it further. We'll keep an eye on it, and many best regards to the person whom this drug helps, obviously. Now, this guy here has already undergone 15 clinical studies with more than 100 patients, and it was proposed for practical use in China. However, as of now, I don't know whether it was officially advised and officially registered as a drug. Now, these guys showed themselves pretty effective against the previous SARS outbreak in 2003-2004, and currently they are advised against the current disease as well. So the drug design is going on, and I hope at some point the specific substances will appear and get approved, but right now it's far from completion, unfortunately. So what's about vaccination? What's about vaccines that some teams already are designing and making? Well, here is the reference to rather advertisements and Science Popular article in Nature Research. This company, I'm sorry, I must modify its name. This is the correct name, GeneScript. It has already developed the test, the laboratory test that detects SARS-CoV-2, and currently now it works on the prototype of the vaccine. Now, here is another interesting publication that came out in Cell journal recently, and it took the authors really a short time to prepare a cell-level publication, which is unbelievably uneasy. Usually Cell is the journal that demands a lot of experiments for the publications that go in, a lot of laboratory work, and indeed this paper does meet all the criterion standards for being published in Cell. And there the authors determines the structure of that virus protein that links to the host cells, the protein that anchors the viral particle on the on the surface of the human lung cell and makes it possible for the virus to get inside the cell. Once again, an interesting publication. Knowledge like this, knowledge of the structure of the exposed virus parts, exposed fragments of virus proteins, can surely help for further vaccine design. You know, if we know what is outside the virus, what the virus looks like from outside, I should better say, then definitely we have a clue how to fight against it, right? Or how to make our immune system to react on it. We know how it looks, in one word, right? We can mimic it somehow for our immune system and make the immune system to respond accordingly, but safer. in future, of course. This is just the preliminary study. Now, these two papers here, what they do, they screen a large list of potential antigens and select those which are theoretically the best out of them. So theoretically the best meaning that they, by modeling, bind the receptors on our immune cells the best, and it means that in isolated immune cells they also experimentally induce the best reaction. And also there is such a thing as innate immunity. In addition to lymphocytes who have random receptors on their surfaces, and those random receptors can bind to at least something outside body, or something that gets in the body from outside. And then since it is since all the receptors that can potentially bind to self-proteins, the proteins of our own body, got eliminated and deleted previously in the cell development, the only variety of these random receptors that remains in the body is something that binds to non-self, right? So this is how the adaptive immunity works. However, in addition to these cells, there are innate immune cells, and those guys are tuned for being sensitive to some pan-virus or pan-bacterial patterns, something that is present on large cohorts of pathogens and what is conservative across this cohort of pathogens. And for inducing robust immune response, it's better to excite both of these branches of immunity, and the researchers are trying to find what combination of antigens can both stimulate the lymphocytes, the adaptive immunity cells, with the random receptors, in total specific to just something generic, non-self for the body, and in addition to this, to excite the innate immunity cells which are sensitive to pan-pathogen patterns. Alright, so interesting papers. I recommend to go through them. They can give you some insight. This company here started clinical tests just a day ago, clinical test of its own vaccine. We'll keep our fingers crossed. Let's hope something comes out of it. The more studies like this we have, the farther they approach us to the better outcome, I hope. Now, this big table summarizes some challenges and obstacles and some hints as well for further vaccine designs. In case of different strategy of vaccine design, in case of different vaccine composition. So, for example, if you have vaccine based on inactivated virus particles, these are the advantages, these are the disadvantages. If you have the vaccines based on subunits, here they are. The little numbers here represents the references of original studies within this paper, but I believe this paper should be downloadable for free, as majority of the papers that I cite here. Even the paper in Cell, this Walls and co-authors, it was available for me to be downloaded for free. I don't know why. Maybe because of the epidemic situation in the world, or maybe because the authors paid the journal to make their research free, whatever. But just you always can hit a pause button and read carefully what this table says. So what's gonna be next? The vaccine, as I said, is being developed, several vaccines. So what's about the tests? How do we detect this virus in supposedly infected individuals? Well, at least two huge biochemical consortiums already provide test kits for detecting this infection, Roche and Thermo Fisher. Those kits are not something that you can buy, for yourself and use at home. Rather, it's the kits for medical labs. But at least in Europe and the United States and countries with advanced medicine and medical care, hospitals already have these or similar test kits and use them routinely and robustly. Those ones are based on PCR, polymerase chain reaction, and they detect the nucleic acids of the virus, specific RNA that is present in that virus and nowhere else. Okay. However, until they come up with effective treatments or vaccine, we will have to just do something by ourselves, using regular genetic measures of precaution. And for this, please visit the webpage of CDC, visit the webpage of World Health Organization, read what they recommend, how to behave in order to avoid being infected or minimize the risk, I should say. It seems like most of commonly used disinfectants successfully destroy SARS-CoV-2 viral particles. Ethanol, bleach, heating, whatever. Even applying regular soap effectively washes it off your hands. The strategy of social distancing is recommended and is suggested helpful, and indeed it has deep rationale between it, since the virus cannot really travel by itself for long distances. The risk of getting infected when you stand straight next to the infected individual is higher than when you are a little bit away. Keep it in mind. I'm just saying it's all per CDC. It's not something that I thought up. So I'm just a transmitter of the commonly approved information to you. The last paragraph here, avoid touching your face when at risk. Well, it seems like our skin is a pretty good barrier. Not ultimately good, but still a relatively good barrier for the virus. It doesn't penetrate through the skin very easily, unless the skin is damaged, unless you have wounds or scratches or cuts or anything. But how the virus can go inside the body is through your eyes, through your mouth, nose, through exposed mucosal surfaces. So you can have some traces of this virus on your skin, intact skin, but if you touch your eyes with that virus or touch your nose, then you're pretty likely to get infected. And of course, it doesn't imply that you can safely get yourself coated with the virus and start walking around. No, please take measures, as I said, of removing it from your skin. But the most of gate for the virus is suggested to be open mucosal surfaces. Okay, enough of this. I think you understood everything I wanted to say about this slide. So what's going to be next is the epidemic kinetics. And we will examine it in short in two alternative polar situations. What happens if the population is freely mixing, if people are freely traveling to unlimited distances from the infected zone to uninfected zone, and what happens in quarantine. The real truth is somewhere in between them, and also the models that I'm going to show you do not capture many significant medical facts. However, as an estimation, as the model that qualitatively show how the disease can develop, I guess they are well, relatively useful, at least by their simplicity. At least they are useful because they are simple and capture some major characteristics of the epidemic-kinetics. So what happens if the population is freely mixing? Well, then let's denote time as t, let's denote x as the number of infected people. dx over dt is the velocity with which the number of infected people varies through time. And since there is some probability, some constant probability that an infected person can recover and get healthy again, there is this term in the equation that depicts the influence of the recovery into the total speed, the total velocity of the number of infected people, and I call it a recovery rate. And it's basically just it's basically just number of infected people multiplied by some positive constant number. The constant, not formally likelihood, but the constant rate with which relative rate. This is the right word. Constant relative rate with which the infected people get healthy again. But also, the infected people can infect other people, and the infected person can travel outside the infected zone, being surrounded by healthy individuals, and then spread the disease, and one person can infect two can infect four can infect 8, and so on. And this is denoted here by, again, the number of infected people multiplied by a positive constant factor, which represents the infecting rate. And then what we can do, we can just move the x out of parentheses here, and this equation has a trivial, very easily obtainable solution, and it basically stands for exponential growth. If the relative infecting rate is greater than the relative recovery rate, if this factor here is positive, then the number of infected people keeps growing and growing and growing, and formally, mathematically, it grows ad infinitum. It grows with no stop forever. However, at some point, it just simply hits the number of total number of people in the population, and then everyone gets infected. So this thing basically says, if the population is freely mixing, then formally this function never stops growing. That's what it says. Even though there is some recovery rate is assumed in this system, no matter what this recovery rate is, as soon as it is smaller than the infecting rate, the function will grow and fill up the entire population, the entire number of uninfected people. All right, so that's pretty scary. However, what can happen, again, at the very basic level of approximation, at the very formal, in very formal examination, what can happen if the quarantine is applied? Well, then the disease would spread only through the border of the infected zone. Assume we canceled all air flights, all distant travels by trains, buses, whatever, understand the way how a healthy individual can get infected is only if he or she shakes the hands with the infected individual who is sitting inside the infected zone. So there is the surface of the earth, and there is some patch on it, some city probably, or a village which is infected, and then nobody travels outside, but if you approach close enough, you can get infected from the people inside. And then the recovery term in the equation will stay the same. It's L times X, nothing changes, but the disease spreading term is now proportional not to X itself, but to the square root of X. And R here stands for also some positive constants. This equation is also pretty easily solvable. It has a solution, and not as trivial as the previous one. You have to work a little bit to solve it, but it's pretty easy still. And here is the solution. Here is the solution, and here is the graph for this solution. So first of all, this current solution has two dramatic differences with respect to the previous one. It does not grow forever, formally. It always plateaus. Now, in the real situation, of course, this level of this plateau may be so high that it is higher than the entire number of people in the population, entire number of people on Earth, and for our purposes, the disease will anyways take the entire population. However, it all depends on these constant parameters. If the values of these parameters are such that the plateau is lower, then the quarantine itself should be enough to limit the number of infected people in the end at infinite time at some constant finite level. So that's one interesting fact, and the second interesting fact is that here at the beginning, the initial growth of the number of infected people is parabolic but not exponential as before. And from the school course of school classes of mathematics, high school classes of mathematics, we know that parabola grows slower than the exponential function. So these two simple and probably oversimplified models already give an understanding of how the quarantine is important and how social distancing is important as well, because social distancing is a sort of quarantine, if you will. All right, that's a simple way of thinking about epidemic kinetics. What's about the more profound and more elaborate models? Well, there are a few. For example, this paper uses a huge system of equations and a lot of computational power, and they came to the conclusion, they sort of found out that it is indeed likely, and it does not contradict to the model, that the reason why the epidemic in Wuhan, China, where the disease first started, the reason that this epidemic slowed down was indeed attributed to the quarantine. Just introduction of the quarantine, according to the model, leads to similar effect as was observed in Wuhan. Okay, as we know now, the center of the disease spreading moved to Italy, and the publication here a sort of attempt to figure out the roadmap, the number of guidance for future action, what to do in Italy if we assume that the initial growth is indeed exponential and not parabolic. How the disease will spread, if the disease transmits not only through the border but in a well-mixed population. All right. Well, I'm sure there is a lot to say more about this situation. However, the state of things changes so rapidly that whatever I say now can be outdated already 2 days later or a week later, whatever. This is why I preferred to pronounce here only the most conservative observations and conservative data that are unlikely to change significantly. However, obviously that's life, they also can change. Now I'm transiting to the list of references, and again, as I said before, you're welcome to press your pause button and copy the entire these things, the entire these rows, search for them in the internet and read the materials, because I know something in my presentation here could be misunderstood or could be unclear. I was in rush when I was preparing it. You see, I even did not translate, accidentally did not translate some of the titles, which I again apologize for. Well, I hope it was not boring, and maybe see you again. Goodbye everyone, thanks for listening.